Your retina is built backwards. So why can you see? Most cameras place the sensor directly behind the lens. Light enters, strikes the detector, and an image is formed. Your eye does something that sounds much stranger. Light passes through layers of living nerve cells before it reaches the rod and cone photoreceptors that actually detect it. The photoreceptors point away from the incoming light toward a dark sheet of cells at the back of the eye known as the retinal pigment epithelium. That is why the vertebrate retina is often called backwards or inverted. It sounds like a design mistake, yet this tissue can detect single photons, distinguish millions of colors, and give us extraordinarily detailed vision. So why is the retina arranged this way, and how does it work so well? This is Biology Through Time, where we explore the discoveries, mistakes, and biological mysteries that shaped our understanding of the world, and the technologies that could shape what comes next. The comparison with a camera is useful, but it can also mislead us. A camera sensor is an electronic surface that can be manufactured, placed in position, and replaced. A retina is a living neural tissue. It needs to develop, process information, maintain itself for decades, exchange nutrients, control its chemistry, and remain connected to the brain. The lens and cornea focus light onto the retina, but the retina is not simply a passive screen. It performs the first stages of visual computation. It detects contrast, separates light from dark, contributes to color and motion processing, and compresses an enormous stream of information before sending it down the optic nerve. For much of history, people did not know whether vision entered the eye or somehow emerged from it. Some influential ancient thinkers proposed that the eyes emitted rays that touched the world. Others argued that light or images traveled inward. Both ideas were attempts to explain something deeply puzzling. Vision feels immediate, even though the eye is only receiving light. The decisive shift came when optics became mathematical. Instead of asking what vision felt like, investigators traced the path of light through transparent materials. That created an uncomfortable result. A lens does not project an upright picture. It bends rays so that they cross, and the image lands upside down. In 1604, Johannes Kepler described the eye as an optical system that forms an inverted image on the retina. This separated two questions that had often been confused. Physics could explain how light formed an image inside the eye. The brain, not the lens, had to explain perception. We do not experience the world upside down because the brain does not inspect a tiny photograph sitting at the back of the eye. It interprets patterns of neural activity using movement, touch, balance, memory, and information from both eyes. Kepler's insight helped transform vision from a philosophical mystery into a problem that anatomy, physics, and neuroscience could investigate together. Now look at the tissue itself. The retina contains several layers of cells. Closest to the incoming light are ganglion cells and their nerve fibers. Beneath them are networks of amacrine, bipolar, and horizontal cells. Deeper still is the outer retina with rods and cone photoreceptors. Their outer segments, the structures contained in the light-sensitive pigments known as opsins, sit beside the retinal pigment epithelium, or RPE. So photons travel inward through the neural layers to reach the photoreceptors. The electrical information then travels in the opposite direction, from photoreceptors to bipolar cells, then to the retinal ganglion cells, whose axons gather together to form the optic nerve. Light goes in, information comes back out. This arrangement is more than a bundle of wires. The retina begins analyzing the visual scene before any signal reaches the brain. Horizontal cells compare neighboring photoreceptors and help emphasize contrast. Bipolar cells divide signals into pathways that respond differently when light increases or decreases. Amacrine cells shape timing, movement, and interactions between channels. Ganglion cells do not simply report the brightness of individual pixels. Different ganglion cell subtypes respond to features such as edges, motion, direction, color contrast, or changes over time. The optic nerve therefore carries a highly processed description of the scene, not a raw photographic copy. The retina is best understood as a thin extension of the brain placed inside the eye. At the deepest neural layers of the photoreceptors, rods are extraordinarily sensitive and dominate vision in dim light. Cones operate best in brighter conditions and support color and high acuity vision. Both use stacks of membrane packed with light-sensitive proteins. When a photon is absorbed, it changes the shape of a light-sensitive molecule and triggers a biochemical cascade. Unlike many sensory cells, photoreceptors reduce their neurotransmitter release when light activates them. That altered chemical signal is interpreted by the retinal network. The outer segments must operate continuously while being exposed to light and high metabolic stress. They are highly specialized, but also fragile, which is one reason their position beside the RPE matters so much. The retinal pigment epithelium is a single layer of pigmented cells, but it performs an extraordinary list of jobs. It absorbs stray light, it transports nutrients and waste between the retina and the blood supply beneath it in the choroid. It helps recycle the vitamin A-derived molecules needed for vision. It forms part of the outer blood-retinal barrier. And every day it engulfs and digests worn portions of photoreceptor outer segments. Photoreceptors continually renew those light-catching membranes, so the RPE acts like a maintenance and recycling system. Placing the outer segments directly beside the RPE and the underlying choroidal circulation gives them access to the intense support they need. The backwards orientation is therefore tied to a vital biological partnership. But light still has to cross the inner retina. Why is it not scattered into a blur? Part of the answer may lie in Müller glia. These long support cells in yellow span almost the entire thickness of the retina. Experiments have shown that their shape and optical properties can provide relatively low scattering paths through retinal tissue, leading researchers to compare them with living optical fibers. That does not mean every photon travels through a perfect biological cable, or that Müller cells solve every optical problem. The retina also benefits from transparent cells, ordered tissue, and specialized anatomy. It is also incredibly thin at about 0.5 millimeter in humans. But the finding about Müller glia shows why calling the retina simply badly wired is too crude. Living tissue has evolved ways to work around its own architecture. The most dramatic adaptation is found in the fovea, the small central region within the macula used for our sharpest vision. Here, the inner retinal layers are displaced to the sides, creating a shallow pit. Light gets a more direct route to a dense mosaic of cone photoreceptors. The central fovea also lacks the blood vessels across other parts of the inner retinal surface. These features reduce scattering and improve spatial resolution precisely where detail matters most. When you read a word or study a face, your eyes constantly move so that the most important part of the scene falls directly onto this tiny region in the center of the retina. Peripheral vision is excellent for detecting movement and navigating space, but the fovea supplies the fine detail we experience as a clear visual center. The inverted arrangement also creates a genuine compromise, the blind spot. The axons of retinal ganglion cells run across the inner surface of the retina, converge at the optic disc, and then leave the eye as the optic nerve. Where they exit, there are no rods or cones. Each eye, therefore, has a small area of the visual field that it cannot detect. You rarely notice it because the blind spots of the two eyes occur in different places, and the visual system uses surrounded information to create a stable perception. But you can reveal it with a simple test. Close one eye, stare at a fixed mark, and move the page until a nearby dot disappears. The missing information was always there. Another compromise is the blood supply on the surface of the retina. Retinal neurons are extremely metabolically active, so they need a constant supply of oxygen and nutrients. In much of the retina, that support comes partly from blood vessels that run through the inner retinal layers. These vessels are essential, but they also sit in the path of incoming light and can introduce some scattering and shadowing. The visual system normally compensates for these fixed shadows so effectively that we do not notice them. Under unusual conditions, however, People can sometimes perceive their own retinal vessels as branching dark lines. The fovea avoids this problem by remaining largely free of inner retinal vessels, helping preserve the clearest possible central vision. So why did evolution not reroute everything? Because evolution does not design organisms from a blank sheet of paper. It modifies inherited developmental programs. If a structure works well enough for survival and reproduction, later changes tend to refine it rather than rebuild it completely. The vertebrate retina arose from neural tissue that folds during embryonic development, and its orientation is embedded in that developmental history. Over immense periods of time, evolution improved the system with transparent neural tissue, retinal processing, Müller glia, a specialized RPE, and, in primates, a fovea. The result is not the simplest imaginable eye. It is a highly capable eye assembled by modifying what previous generations already possessed. The octopus offers a fascinating comparison. Cephalopods independently evolved camera-type eyes with a lens, iris, and retina, but their photoreceptors face the incoming light and the nerve wiring remains behind them. Their optic nerves do not have to punch through their photoreceptor layer, so they lack the same kind of blind spot. It is tempting to declare the octopus eye the winner, but the two systems developed from different tissues through different evolutionary histories, and each works within the biology of its owner. Evolution can reach similar optical solutions through very different construction routes. The comparison does not prove that the vertebrate retina is uselessly designed. It shows that history constrains which solutions are available. The retina's close cellular partnerships also create vulnerabilities. In age-related macular degeneration, or AMD, dysfunction involving the RPE, Bruch's membrane, inflammation, and the underlying blood supply can ultimately damage central photoreceptors and lead to a loss of vision. In retinitis pigmentosa, inherited mutations progressively disrupt rods, cones, or the cells that support them. In glaucoma, the vulnerable cells are retinal ganglion cells and their axons in the optic nerve. These diseases are different, but they reveal a common principle. Vision depends on an ecosystem. A photoreceptor cannot survive merely because its light-sensing proteins still work. It also needs energy, waste removal, structural support, intact synapses, and a functioning pathway to the brain. Break one critical partnership and the entire visual system can fail. Can we repair the retina? That complexity makes retinal disease difficult to treat, but it also creates several points of attack. Gene therapies can replace or modify defective genetic instructions in selected cells. RNA-based treatments may alter harmful gene expression. Cell replacement approaches aim to restore RPE cells, photoreceptors, or retinal ganglion cells. Retinal organoids grown from human stem cells allow researchers to study development, test medicines, and model inherited disease in tissue that resembles part of the human retina. Other teams are exploring electronic implants, optogenetics, and methods to reactivate regenerative programs. None of these approaches is a universal cure just yet. The retina is a circuit, a tissue, and an ecosystem. Successful repair may require preserving or rebuilding several of those levels at once. So is the human retina built backwards? In a narrow geometric sense, yes. Light crosses neural tissue before reaching the photoreceptors that are light sensitive. But that description leaves out almost everything that makes the retina remarkable. Its orientation places photoreceptor outer segments beside the RPE and a major source of metabolic support. Its neural layers process visual information before it leaves the eye. Müller glia cells and the fovea reduce optical penalties, and the brain compensates for gaps such as the blind spot. The retina is not a camera sensor installed by an engineer. It is living brain tissue shaped by hundreds of millions of years of evolutionary history, ingenious, vulnerable, and still only partly understood. In next episode, we will discuss why some animals can regenerate the retina while humans largely cannot.